how the virus can replicate and the efficiency of replication. And so we assayed this by plaque assay, and when we look at the blue here is wild type, the red is your back knockout, and the brown is the back-to-back double knockout, we see that they're all relatively similar in their maximal viral titer that's produced. Although the back knockout cells, this red one, replicates a little faster in the early stages of infection, the overall total viral titer, the maximal viral titer that's generated, isn't so far off from the wild type. But when you look at these back knockout cells that are dying by autophagy instead of apoptosis, what you see is a 2-log drop. This is green line here, a 2-log drop in viral replication. And so there's something going on here that we wanted to explore a little further and answer why is it that these cells here, the back knockout cells, have such a problem producing infectious virus. And so we wanted to make sure that this effect we see with the plaque assay is not due to some remnant of the knockout conditions or the formation of the knockout cells. And so what we did is we took wild type and Bax knockout cells and we reconstituted Bax back in. And what you see here is the fold increase of replication over the native cells that don't have Bax reconstituted in. And so with the wild type that already express Bax, when we topically express more Bax, we don't see much of a change at all, maybe a slight increase in the amount of replication. But when you add Bax back into the Bax knockout cells by transfection, what you see is an over 5-fold difference, an increase in the amount of replication, indicating that this is indeed a function of the Bax knockout and not some other aberration of the cells. So the next question we ask, plaque assay is an indicator of viral infectivity that's being released. We wanted to see what happens with the amount of actual virus that's being released. And so what we did was we extracted RNA from the supernatant and we looked at the amount of viral RNA after 24 hours, 12 and 24 hours, that's being released from these cells. You can see here in the wild type versus all 3 knockout conditions, the 3 knockout conditions all have about the same as double the amount of virus coming up. This indicated to us that this data here, the infectivity data, is not a function of the amount of virus that's being released, but a function of instead something going wrong with the assembly or the maturation of these viruses, therefore affecting the assembly, infecting the infectivity. And so when we looked at NP retention, this green right here, NP, remember, is that protein responsible for delivering the genomic segments from the nucleus out to the cytoplasmic assembly centers. We see in the wild type, the back knockout and the back double knockout, no real nuclear staining here, although if you look at the quantitation, we do see some NP retention inside the nuclei of these cells. However, in the back knockout cells, what we see is 100% of the cells that we assayed had severe NP retention inside the nucleus. We confirmed this by nuclear fractionation of infected cells, and we took the cytoplasmic and the nuclear fraction of each cell type after infection, and you can see here that we indeed have quite a bit of nuclear retention of this NP protein in the cells. And this indicates to us that the defect in infectivity of the viruses is at least in part due to the ineffective assembly of the viruses because we have these genomic segments stuck inside the nucleus and not at the assembly centers in the cytoplasm where they need to be. So in summary, influenza A induces apoptotic death in our fibroblastic system. It also, this death after ZVAD appears to switch from apoptosis to autophagy in nature. BAC is potently downregulated while BAX is activated during flu infection. And this death, influenza A-induced apoptosis, is severely inhibited in BAX knockouts, which instead demonstrate features of autophagy. And the last, manipulation of viral titer appears due to problems in influenza A NP transport, resulting in immature or not fully assembled virions being released from these cells. So influenza A is killing by apoptosis, and the summary is basically a repeat of what I just said, so I won't repeat it. The next project we have is flaviviruses. There have been several reports, and you heard one earlier today, that flaviviruses kill by apoptosis. And so when we started this project, we were honestly looking to compare how the influenza A apoptotic effect and the flavivirus apoptotic effect actually compare. What we found after several tries, well, first of all, let me get to here. Flavivirus, in the first place, represent several medically important viruses across the world. Several million people a year are at risk for being infected with flaviviruses. Some of these have no treatments, no cures, no vaccines. And they include Japanese encephalitis virus, which kill about 90,000 people a year. And even if you survive, they experience lifelong neurological effects. The yellow fever virus is another big one, with a 50% mortality rate, killing about 100,000 people a year. We just got a grant, so I'm really, really happy with the dollar signs. The ones we studied in particular are dengue fever virus, and this is a globally, equatorially endemic virus, about 50 million cases a year, and about 500,000 of these cases result in a hemorrhagic fever, which is not nice at all. And about 5% of these victims die per year. And last year in Rio, there was a big epidemic. They killed over 500 people in the span of a couple of months. And with this dengue fever virus, because we don't understand the biochemistry behind these viruses, there is no cure, there's no treatment, there's no vaccine for these viruses. So it's really important to understand the biochemistry behind this. And as a parallel study through all these, you'll see that we have Modoc virus data as well. And Modoc virus has been proposed in vivo by Leyson's group in the Netherlands as a very good, safer model for dengue infection in the lab. So the first question we ask is, do flaviviruses affect cell survival during infection? And so after try after try after try, well, we found that these viruses in our system in fibroblastic cells don't kill the cells. And this gets ridiculous because even at an MOI, a multiplicity of infection of 1,200 viruses per cell, which we'll never see in a physiological situation, we still don't see death. And even at very, very late time points, 288 HPI, that's about 2 weeks, we don't see any death whatsoever going on. And this observation actually goes across several different cell types. We took Swiss Webster MEFs, which have a deletion in their flavivirus resistance gene, which makes them susceptible to flavivirus infection, and no death there. C57 blacks, which have the flavivirus resistance gene intact, no death there. MDCKs, which are dog kidney, no death there. And Vero cells, which is the classic in vitro system for dealing with flaviviruses, we don't see any death here as well, even up to 144 HPI. And so the obvious question is, are these viruses actually infecting the cells? And so what we did was we took a dengue envelope protein-specific antibody and looked for antigen inside infected cells. And with both Modoc here and with dengue, you can see in the perinuclear region, which is where these viruses are supposed to be, right at the ER, we see significant amounts of staining there. And when we titered these viruses and the release of the viruses coming up, by both RT-PCR here and by FACS for both Modoc and for dengue virus, what we see is these viruses are released in our hands similarly to other published reports from other different labs. So yes, the virus is infecting the cells, and yes, it is able to replicate in these cells. So something else is happening here. And so the next question we asked was, if the cells aren't dying, well, maybe they're actually protecting against death. And there's a couple of published reports of West Nile virus that indicate that, yes, some flaviviruses do indeed protect against death. And so we tried this in our hands in our lab with Modoc and dengue virus. So we started with Modoc virus, and you can see with camptothecin, which is a DNA damage inducer, staurosporin, which is a kinase inhibitor, cyclohexamide, which is a protein translation inhibitor, and also Influenza A, which I just showed you induces death by apoptosis. We see a marked reduction in the amount of death after infection. So we infected the cells with the Modoc or dengue virus for 24 hours, then we treated with a death inducer for 24 hours, and then we assayed for death. And you can see between a 15 and 40% reduction in the amount of death after infection, depending on the inducer, but in every case, we do see protection against death. And for Modoc virus, we have our data at 48 hours, and a late time point, 144 hours. And with dengue 2 at 44 hours, we see very, very similar reductions in the amount of cell death versus all 4 of these inhibitors. And so we wanted to make sure that our system was medically relevant, and so we went for some human cell lines, HeLa, which is cervical carcinoma cells, which is the blue, and 293T, which is human kidney cells. And again, we see protection against death with both Modoc here and with dengue. And just to back up our data, we also did primary Swiss-Webster maps straight from the mice. And again, we see similar levels of protection. About 15 to 30% is usually what we wind up seeing. And so the next question is, how do these viruses do this? And so there have been published reports of other flaviviruses manipulating the PI3 kinase pathway. And so what we did was we decided we'd investigate the PI3 kinase pathway by blocking with Wortmannin, which can block both mTOR activation here and also autophagy. And so we again did the LC3 GFP punctation, and in our hands with the mock, you see nice diffuse LC3 distribution, again indicating inactive cytoplasmic LC3, but with both Modoc virus and dengue virus, you see these bright punctate dots suggesting LC3 translocation to autophagosomes and an autophagic response. However, when we treat these cells with Wortmannin, this autophagic response goes away completely. And so what we see quantitation with just the virus, close to 70% of the cells are autophagic, but with this PI3 kinase inhibition, it goes down to nothing. And so our next question was, if the autophagic response is being so greatly affected by PI3 kinase inhibition, what will happen to this protective effect? And so we did the same experiment, only this time we treated with camptothecin again. And you can see here, when you have just mock with CPT, significant levels of death, over 50% after 24 hours. When you infect with Modoc virus and hit it with CPT, we see a close to 30% reduction in death. But when you block the PI3 kinases with Wortmannin, that protection is completely eliminated. And so flavivirus infection does not cause cell death in several fibroblastic cell types. Post-infection protection against death is induced against several inducers is observed, and the autophagy response that we see is blocked by PI3 kinase inhibition, as well as the protective effect we see after infection. And so the last question that I'll be talking about here today that we ask is what viral genes are actually responsible for this cell survival effect during flavivirus infection. And so the flavivirus genome consists of 10 different genes. It's a linear genome. This linear genome is a positive sense genome. So what happens is it encodes, it gets into the cytoplasm, and it immediately starts getting translated by ribosomes. We form a 10 kilodalton polyprotein, which then weaves its way in and out of the ER, and then the individual viral genes are clipped from the polyprotein, first by cellular proteases, and then the NS3, NS2B viral protease is cleaved, which then goes back and clips the rest of the viral proteins. So what you have at the end from that single polyprotein is 10 different viral proteins. And so we assayed 3 viral proteins in Modoc to start, NS2A, NS4A, to which there has been no published function that we found in the literature, and NS4B. And you can see the empty topo virus, we're looking at.
So what do you think is the physiological role